Well, friends, I would like to welcome all of you to this program about COVID-19 vaccines and immunity. I am indebted to the Baha'i community of Surprise that took up this effort to bring us together and have this conversation. This conversation has a scientific flair, even though the Baha'i community has sponsored and put this event together. The views that are expressed here are all based on my research and my understanding of the literature and the status of the pandemic, and are not reflective of the opinions of the Spiritual Assembly of the Baha'is of Surprise or that of the Baha'i faith. This is a scientific presentation, and it is meant to help with our understanding of where we have that has been given to me to share with you some of the findings on the basis of my medical training and my understanding of this pandemic. There is a lot of consternation, there's a lot of anxiety, understandably, there's a lot of lack of trust in the process, in the discovery and the development of vaccines specifically. So I will focus specifically on vaccines for about half the presentation, and then I'll focus a little bit on immunology for the latter half of the presentation. The entire presentation is allotted about 1 hour of time, so I am not going to take a lot of your time. I'm going to probably briefly skim over some of the slides just so that we can have more time in exchanging, because there's a lot of material out there. There is also a call by the CDC on medical professionals to promote vaccination as a way to shorten the lifespan of this pandemic. So I, as a physician, have answered that call and welcome these opportunities to have these conversations after having reviewed so much of the data that is now available. So as you know, there are, when we want to go through bringing any medication to the market, it has to go through preclinical trials, which typically involves animal studies and laboratory studies. And then we go through clinical trials, which have phases 1, 2, and 3. And I have a slide that will go over as to how that happens. And then after that, phase 4 essentially is once the product has entered the market, there is still a reporting and safety mechanism that continues to gather data and refine our understanding of safety and efficacy. For any medication, the first thing that we want to know is this safe? Meaning the first axiom of medicine being first do no harm, which is in the Hippocratic Oath, or the ethical principle of non-maleficence, is very important to us. So the first thing to do is, could this thing be more harmful than it can be good? So that's where safety comes in. And then the second issue is, does this product do what it is purported to do? Will it particularly work for the intended consequence that we're seeking? So for example, if it's supposed to bring you immunity against the virus, does it do that? If it's supposed to lower your cholesterol, does it do that? And the comparison is always done with a placebo, which is done in a control group. So typically phase 3 trials involve a significant number of volunteers that come in and in the tens of thousands, and then they are looked at as far as half of them will receive the actual therapy, the other half do not. They are blinded to the fact as to whether they received the real medication or if they received placebo, which could be a host of things. In a lot of these studies, the placebo was a typical pneumovax, just a vaccine for your run-of-the-mill pneumococcal pneumonia. And the investigators are also blinded to this matter. They don't know who has received what. And they follow these patients for a period of time. And once they gather their data, they unblind the study. So the highest quality of data that we have in medicine is obtained through what we call randomized controlled trials.
Yeah.
If you would be so kind to mute yourselves so that we do not have multiple voices coming in at the same time, I will allot plenty of time for us to exchange. But I wanted to say that the current vaccines that now we have under development and in the market all have gone through this rigorous analysis of a randomized controlled trial, which is the gold standard of evidence in medicine. So the 4 phases are phase 1 of clinical trials. This is, I'm skipping the preclinicals on the animals and coming to clinical trials. It's phase 1, we just want to get a sense of the product somewhere in the double digits, somewhere between 20 or 100 healthy individuals are going to come just to see if there are any serious side effects, if there's anything very significant. This phase is usually very short. You get past it very quickly. Then you go to phase 2, where several hundred volunteers come in, and you start looking at still the safety, but now you're starting to look at some laboratory evidence of, in the case of vaccines, immunity, for example. Is there any immunity that is starting to develop? And immunity is typically measured in the form of antibodies that are formed in the body. And you get to compare these antibodies to those who have had the disease itself, in this case, COVID-19, and see if these antibodies are similar so that you can have a window into prognosis as to whether the patients would actually be protected if they were to come in contact with the virus itself. Phase 3 is typically in the tens of thousands of volunteers, and this is when we're looking at any minor side effects or any rare side effects that may not be picked up with a few hundred in phase 2. And of course, we also want to understand efficacy. And I'll get to how efficacy on vaccines is particularly defined. And in phase 4, the vaccine is approved for mass use, but still data continues to be collected on people. For example, in America so far, we've had over 20 million Americans who have been vaccinated, and we get to study the side effects that they have, and there are applications that they can report through text message conversations and interviews if they have any side effects, so that the mass of data continues to come in and be recorded. So, so that's phase 4 really. And you should consider that typically what happens with the FDA is that a vaccine needs to get licensed, but in case of emergencies such as a pandemic that we have on hand, there's another pathway that allows for a sooner appearance on the market of these medications when you weigh the risks versus benefits, and that is called the EUA, which is Emergency Use Authorization. So the current vaccines that we have on the market are not licensed as such. They have received the EUAs from the FDA. There are 2 of them that have received this. One of them is Pfizer-BioNTech. It is important, I know everybody says Pfizer, but I am going to be a little bit of a stickler and say, I encourage you to include BioNTech specifically because the brains behind the development of this particular vaccine really comes from BioNTech, which was a cancer company that had a lot to do with immunotherapy against cancers, and they had developed the mRNA technology within them for cancer care, and they ended up venturing into the pandemic business. And so Pfizer did not have such platform, but Pfizer, because it was a much bigger company, had the machinery and the infrastructure to partner with BioNTech to bring this vaccine to the market. They reported 95% effectiveness. We will talk about the definition of efficacy in just a little bit. We will also talk about Moderna that came virtually at the same number of 94.5% efficacy. And if you want a little bit of a comparison, the seasonal flu shot that you receive every year is somewhere between 45 to 55% efficacious. And you go and religiously and willingly receive it most every year. And so just to give you an understanding, the more efficacious the vaccine, the sooner you will get to the concept of herd immunity. The less efficacious, the more of the population you need to vaccinate as far as percentages to get to herd immunity. And we are going to talk a little bit about what herd immunity actually means. These two vaccines in particular that are now in North America approved for use, they both utilize mRNA technology. mRNA technology is a relatively new concept in vaccinology. It was not anything, we have never had an mRNA vaccine before these vaccines, but they are also ones that, because of the simplicity of how they are made, were the soonest to market, because there were other kinds of vaccines where you use viral vectors, for example, non-disease causing viral vectors, such as the AstraZeneca vaccine, that are actually a little bit harder, more cumbersome to make, whereas these are easier to mass produce. And so in that sense, there is an advantage to these. But there are also some logistical challenges with mRNA vaccines, such as the need to keep them in very significant sub-zero temperatures to keep them stable in solution. Okay, so these have been tested in diverse populations. A lot of these have references on the bottom from the CDC. If you wanted to get a sense, so much of what I'm going to focus on today is so that I give people the confidence of knowing that the science behind these vaccines is sound. The Pfizer BioNTech had nearly 44,000 volunteers at 150 different clinical sites. The number of clinical sites is also important, because if you only use one or two hospitals or cities and just give one group of people in charge, if they are corrupt and they want to cook up their data, it's much easier for them to do that. When you grab it from multiple sources and diversify, then you actually are far more likely to trust the data that comes in. So 150 different clinical sites, nearly 44,000 volunteers in 39 states in the US, with a diverse racial distribution and age distribution. You can see them on this slide. I'm not going to go over all of them. Moderna had 30,000 enrolled at 89 clinical sites in 32 different states, and had also a good racial distribution, because we know that certain races, for example, the African American and people of Hispanic descent, ended up having more significant issues with this virus. And it was very important to ensure that they are also protected. And so the data came in, and the data was very significant for a very good amount of protection. There's also further data that I was reading today that did not make it to this presentation, that Israel, which is now leading the world in the total percentage of citizens that have been vaccinated, they are now approaching 30% of their population, not only have already started to see a decrease in new cases, but of the groups that have been vaccinated, which are close to 2 million, there's only been 68 cases of people who have contracted coronavirus. And within that group, the ones that have contracted it have had it very mildly. So efficacy is not 100%, with very, very small amount, something like 0.1% of that population that has been vaccinated with the 2 doses actually ended up getting the virus. This one is talking a little bit about the mRNA technology. And what I wanted to say is that mRNA is essentially it's a recipe. That's all it is. It's a recipe. So in the same way that if you want to make peach cobbler, you can get the recipe. There is a particular recipe for the spike proteins, a couple of very significant spike proteins that the virus happens to have, by which it can attach to our cells and inject its genetic materials into our cells. And if the virus were to lose those spike proteins, or if we were to inactivate those spike proteins, the virus, if it entered our body, it could not make us sick. These spike proteins have a very high affinity for a particular receptor in our cells that we have in a variety of our tissues, not just our lungs. We have it in our nervous system, we have it in our livers, we have it in our kidneys, and we have it in our muscles, believe it or not. And these are called ACE2 receptors. And so if we had a way to get this spike protein, this little nubbing on the surface of the virus, inactivated by attaching antibodies to it, we would be very good at invalidating the virus, or basically making it incapable of virulence or causing disease. So mRNA is a recipe for making that particular spike protein. That is all it is. So when they inject it into our bodies will say, huh, this looks like an interesting recipe. The body starts making the spike protein identical to the surface protein of the novel coronavirus. And when it makes it, then the body starts looking at it and saying, well, you're not part of us, because the body recognizes its own self proteins. So when it recognizes it as a protein, it starts making antibodies to it. Once we have significant antibodies, the fact that we get 2 shots, it provides a significant amount of memory in our memory B cells that allows for us to then be able to keep these antibodies and have lasting immunity. Is my voice good? Because for a moment I got to notice that my internet is weak.
Yeah, it went away and came back. Thank you.
We're good now? Okay. So the technology is new, and it has been studied. The mRNA vaccines have been studied for years now on influenza, Zika, rabies, and cytomegalovirus. But the first one to market ended up being for SARS-CoV because of the very significant governmental investment in finding a solution to the pandemic. So about these vaccines, what are some of the side effects? The side effects: fever, headache, muscle aches. But there are no significant safety concerns in the research. And so far, in the millions of people who've been vaccinated, except some negative news that came with the Pfizer-BioNTech vaccine out of Norway, that people who were over 80, who were debilitated and were residents of nursing homes, some of them with the second dose ended up having a significant inflammatory reaction, and a very small percentage of them, something to the order of about 29 deaths, had occurred in Norway. I have not seen any such similar report in the US. There was only one report of a death after a vaccine in the US that came in somebody who had had COVID-19 before, received the vaccine and had a very significant immune reaction, ended up passing away, and that's still being investigated. The CDC is not calling it a vaccine death, but this is after 20 million vaccinations in the US. If you have a fever, you have a headache, you have muscle aches, these are all hallmarks of an immune response in your body. This does not mean that you have gotten the disease. To have the recipe to make a spike protein for the virus does not mean that you have the virus. And so if you have flu-like symptoms, they typically last about 1.5 to 2 days and they go away. They are not the disease themselves, and it's just the immune reaction that you're feeling. You can celebrate that your body can do something immune-wise to make you ready for facing the virus in the future if it were to come across it. So safety has been a top priority. There are various committees and advisory bodies that are receiving data back even in phase 4, which is now, as far as the people who are receiving the vaccinations, and the monitoring continues very significantly. So for those who have a wait and see approach, well, we'll wait to see if our neighbors do it first and they do well. That is not a scientific approach, just to see if your cousins or your parents did well. I think that the fact that we now have over 20 million Americans vaccinated, and there continues to be a very good track record of safety, is really indicative of waiting is not warranted at this time. I think that anybody who is within the priority groups, who is called upon, should embrace this opportunity and see it as a privilege, as I saw it as a privilege, and I have received both vaccinations myself. These are the names of some of the agencies that continue to monitor for safety. I just wanted to mention them: the VA, ERS, and the VSD, and the CISA, and BEST. Those are all following very closely, and the surveillance continues. And for those who think that the issue was accelerated, well, it has to do with government funding. It has to do with prioritizing the review and authorization of the data. It has to do with the fact that we were facing an international emergency. And so nobody skipped the line.
Well, again, thank you so much, Harvey. That was an immense amount of diligence.
Okay. So vaccine confidence, what are some of the things? Only 58% of Americans say that they would receive the vaccine. What are some of the reasons why people would not do that? Well, they wonder if it's safe, how much does it cost, and if there are human experimentations, if there's a microchip in there. And I would tell you that nearly all of you who have smartphones, you already carry a microchip that tells a lot about you to all kinds of agencies that you may not want them to know. The vaccine does not, anything that's liquid is incapable of reporting your location or your needs and wants to any government agency. So those who have worries about microchips being installed into vaccines, or the conspiracies of Bill Gates wanting to change your DNA, or changing, or changing your fertility status, all of these things are complete bogus. You cannot change anybody's DNA with an mRNA vaccine. mRNAs, by definition, cannot enter the nucleus of any of your cells. And as far as cost, the government has essentially said anybody who comes up for their vaccination, if they do not have insurance, they still need to be vaccinated. There is no issue with cost here. And so I don't want things to be barriers to people receiving the vaccine. The hesitancy among healthcare providers, believe it or not, also exists, which is mind-boggling. There were significant nurses, about 1 in every 3 nurses, who said they would not receive the vaccine. And there is surveys that show that, that applies to so many healthcare workers, that about 1 in 3 of them want a wait and see approach or have some significant hesitation to receive it. I work with many of them. When I'm in the operating room, a lot of times I do my own mini survey and I say, how many people have received the vaccine? And typically about half raise their hands, and everybody else has some excuse, which is mind-boggling to me. And so I don't want to talk more about the confidence, but basically on the spectrum, because confidence falls essentially on spectrum. There's refusal on one end and acceptance on the other, and then there is this wait and see nebulous middle ground approach, like, well, I'll do it in the future. And so I think that it is our job as healthcare providers who see the data, who see the efficacy, that's so much better than so many of the vaccines, that this vaccine is more similar to measles than it is to the flu vaccine, for example, that we really have an opportunity to make a dent. And one other thing that I would say is that, have you noticed recently that we keep hearing about different variants, such as the UK variant or the South African variant? Well, why is it that these variants are happening now and they weren't happening before as much? Well, first off, they were always happening, but it is true that they're accelerating because the more number of infected individuals there are on this planet, the more the virus will have opportunities for mutations. And so if we want to stop this and avoid another mutation that could potentially be far more deadly, because coronaviruses have had a history of being more deadly. If you remember MERS from 2016, the mortality rate of that was about a third. 1 in every 3 individuals who contracted that disease, Middle East Respiratory Syndrome, ended up dying. And that was a coronavirus too, from the same family. So these things can happen. So the sooner we get this mass immunization, the sooner we get to control the numbers to such an extent that if the number of infected in the world are less and less, the number of opportunities for the virus to have in mutating itself and potentially becoming more virulent will also be less and less. And this falls a little bit under the herd immunity that we talked about. So the people who vaccinate, I think, should share with their friends and share their experience, because so many of us, instead of looking at statistics, end up looking at our friends. And so I think especially those in healthcare, and I know we have some nurses and physicians in the group, they need to promote this to their own peers and their own patients. This is very important if we want to beat this pandemic. And people in leadership position need to remind their employees to get this. No coercion, of course, but at the same time, I think a reminder and creating opportunities for them to take off from work so that they can go receive the vaccine is very important. Education obviously is very important. The effort that we're doing today, along with the Baha'is of Paradise Valley, who were so kind to put this program together, fall under the same effort to try to engage with our communities and answer their questions and talk to them about the vaccines specifically. I want to talk a little bit about immunity as well before I open the floor to questions and a wonderful exchange with you. I wanted to say that we search for serum antibodies, or you may have heard the word antibody titers, that we end up recording these titers as a means of measuring immunity. What's interesting is that antibody titers after you've either been vaccinated are the highest, or after you have had the disease naturally itself. Over time, your body doesn't continue to make these antibodies. So there's a natural decay curve. So if you look on this curve, you will see that the antibodies over a 12-month period continue to go down. And this is about a third of them is left, typically at about a year. That's kind of a standard thing. For different diseases may be different. But the immunological memory tends to actually stay, meaning that if you were to be challenged with the same virus again, your memory B cells, which are part of your white cells that are called lymphocytes, tend to remember, and this is called an anamnestic response. Amnestic response meaning that you forget, anamnestic meaning that, well, you don't forget. And so they will pump these antibodies back and they will fight. There's also another story here, which is called cell-mediated immunity, which also is important in beating this virus. Not everybody who becomes immune will mount a magnificent antibody response. Nonetheless, because antibodies are easy to measure, we are measuring them and following them as a metric for how long immunity is lasting. But we know that as long as we're detecting traces of these antibodies, the memory is always higher than the level of antibodies that we can measure. So the formula for efficacy, somehow the formula didn't basically show up here. Okay, it did. So basically it's a fraction, right? On top of the fraction, you have the attack rate in the unvaccinated, meaning how likely is an unvaccinated person to get the disease if exposed to the virus? And in similar circumstances, how likely is a vaccinated person to get the disease if exposed to the virus? You deduct those two, and in the denominator, you put the attack rate in the unvaccinated, and you come up with a percentage. And so basically, when we say 95% efficacy for this vaccine, it basically means that people who are vaccinated are 20 times less likely to get the disease compared to people who have not been vaccinated but exposed to the virus. The concept of herd immunity, basically herd means group, and we're talking about the communities in which we live. So we talk about the fact that the people who get vaccinated, they themselves are protected. If you have a significant number of vaccinated people around, but among them there are a few unvaccinated people, they get to enjoy herd immunity, meaning that they're also unlikely to get the disease. Why? Because the virus needs people to replicate. The virus has no mechanism of self-replication. It needs hosts, and you and I are all hosts. And so in order for the virus to replicate, it needs to go from one host to another. It infects one person, and you're catching an Uber, your Uber driver has it, you get it from them, you give it to your spouse, and so on and so forth. And so the more number of people around you who are immune, the less ability the virus has while it's running its natural course of replication in somebody's body before that person ends up infecting somebody else, to have an opportunity to infect somebody else. And that is what herd immunity is. And so with different viruses, it's set at different levels what percentage we need in the population to be immune, either by having gotten the disease itself or by having received immunity through vaccination. Currently, Dr. Fauci's prediction is that about 70% of the population, once they have it, you will see a very significant herd immunity effect, that even the 30% who have not been vaccinated start benefiting from the fact that their neighbors and their colleagues have been vaccinated. The Israeli example is that they've already started to see the beginnings of herd immunity because nearly 30% of their populations are immunized, that their numbers are starting to level off. But once that number gets closer to 70%, it's a game changer for the pandemic. So the mechanisms we talked about, the indirect protection of non-recipients of the vaccine, and the levels, of course, depend on the infectiousness of the virus that we're talking about. So what are some of the characteristics of an ideal vaccine? An ideal vaccine should produce no side effects and should make 100% of its recipients immune. Such vaccine is nearly non-existent. I think the closest thing that we've ever had to such a thing in the history of vaccinology is smallpox. The fact that you probably haven't heard anybody in your family who's ever had smallpox has to do with the success of that vaccine campaign around the globe. Another success story, I think, is polio. And polio is another fantastic story of medical achievement that basically by doing these campaigns around the globe, it is really a relic of the past to remember anybody in the families or in your colleagues that have had polio, for example. On occasion, you get an elderly person who's had post-polio syndrome because when they were a young person, they may have had polio in a weak way. And then and now that they're getting older, they may have some weakness returning to them. So it should provide protection not only against the clinical disease, but it should also provide protection against carrying the disease and giving it to other people, and also against reinfection. A good vaccine should have minimal side effects. We talked about that. And it should give immunity against multiple strains of virus. Viruses do tend to mutate and change, we are a little bit lucky. Coronaviruses, even though they mutate, they are not as sloppy as HIV virus or the influenza virus. So even though they change and everybody talks about these variants, they're actually not that bad compared to the nature of some other viruses that are also RNA viruses, just like the coronaviruses. So in that sense, we were kind of lucky because it's easier to make a vaccine for it. The reason it's very hard to make a vaccine for the HIV virus is particularly because it is so sloppy in its own replication. It's also a retrovirus, which means that it is an RNA virus, just like the coronavirus. And so, of course, the immunity, you want it to be long lasting, hopefully a lifetime, but sometimes, even if you have 1 year or so, as long as during that year you can vaccinate the masses, you get to have a significant herd immunity effect. Also, you don't want a vaccine that will interfere with the immune response of other vaccines that's given simultaneously. And of course, the cost and benefits associated with receiving the vaccine should clearly outweigh the costs and risks associated with the natural infection. So for example, if you look at America, we've already had well over 400,000 deaths, and we've had over 20 million infections that are confirmed, but maybe somewhere close to 80 million people who've been exposed to the virus, but so many of them have not been confirmed. And so the number of deaths is clear. But so far, over 20 million in America who've been vaccinated, the number of deaths is this one questionable death in California of somebody who had previously had COVID and had become a little bit of a long hauler and had been told to get vaccinated just to have a longer lasting immunity. So nonetheless, there is no perfect solution, but comparing the risk and benefits of receiving the vaccine and dealing with the often very minor side effects of the vaccine versus taking your chances with the disease itself, especially if you are older or you have comorbid conditions such as obesity, which over 50% of Americans, I think, or perhaps 46% of Americans are now obese, or diabetes or high blood pressure. Those are all very significant comorbid conditions that could land people in the hospital and, of course, create a significant problem. So I think this is a good presentation as far as the amount of information placed on the table about what herd immunity is, the history of mRNA vaccines, and the efficacy and how it's calculated. If you would all agree, I think we can open the discussion to a question and answer type setup and spend the next 20 minutes doing that. Would you agree, Dr. Hajbande?
Yes, please, but those who have questions, they have to raise their hand, but for me to see their hand, they have to activate their video as well. Otherwise, I don't see anything. So when Bijan and I see the hands, we put them in line for their turn to come. Thank you very much.
Wonderful. So I'm going to look in chat, and if I see hands there in the order that I see them, I'm going to call on them. And my hope is that we can have
We have Mojgan Elston.
Thank you. I appreciate Doug and Bahar's input. Thank you so much.
Mojgan Khanum, you have to unmute yourself.
Thank you. My question is, if you know someone who's had it, at what point they're not contagious anymore, so you can be around them, and about that.
Wonderful. So the answer is very clear for people who have any sort of symptoms. The people who end up being asymptomatic carriers, the answer is not as well known, unfortunately. But basically, the idea is this: if you are 10 days past your initial onset of symptoms, what could symptoms be? It could be shortness of breath, fever, body aches, diarrhea, cough, something that you have that you then get tested, you know you're positive. If you're 10 days past day number 1 of symptoms, and for the past 3 days consecutively, you've had no fever, and you have been off of all anti-fever medicines, such as Advil and ibuprofen and Tylenol, then you are okay to go back to work and to go back to your family and sit at the same table and eat with the rest of them. So it's a 10-day mark. And what's interesting is that at the beginning of the pandemic, there was a question that we should have people tested and to have them be negative before they can return to work. Very quickly, they realized that A, this wastes a lot of resources with testing, and B, you will have a lot of false positives because people may have fragments of dead virus in their body that will still trigger the PCR test as positive or the antigen test as positive, but that does not mean that they're contagious at all. So if you follow the 10-day rule and make sure that they've been free of fever for 3 days, they can come back. But that does not mean that all their symptoms have gone away. It's just that they're no longer contagious because people tend to cough for weeks after. Coughing is not an issue. It's just the fever that has to be gone. And there are also people who have anosmia, which means loss of sense of smell. And this particular concern can last for weeks, for some people up to 6 months before they completely restore their sense of smell. So you are not going to isolate yourself for 6 months. Once you're past the 10-day mark, as long as you don't have a fever for the past 3 days, the days 7 through 10, you can reunite with your family and go back to work.
Thank you, Doctor. There's a question from Shuku about preference of Pfizer over Moderna.
Both these are equivalent as far as efficacy, and they both have the same exact technology. They are made a little bit differently, but they are essentially one and the same. The only difference that I can tell you, and this is a very minuscule difference, there are really 2 differences that I've come across. I'll mention both of them, but I am going to tell you, receive the flavor of the day, whichever one they give you. If they give you an option you want to choose, then you can choose on the basis of this. One is that even though significant allergic reactions, I'm talking about anaphylaxis. Anaphylaxis means you're short of breath, your body blows up in being very red and swollen, and your blood pressure drops and you have a sense of doom and gloom. Okay, that kind of allergic reaction tends to be 4 times higher with Pfizer than it is with Moderna. But what does that mean? For Pfizer, it is 11 per 1 million. For Moderna, it's 2.5 per 1 million. So Pfizer has a little bit, well, it's 4 times as high, which sounds very high, but 11 per million versus 2.5 per million are very, very small numbers still. So I am not going to refuse one on the basis of that sort of statistic. The second thing is that the people who have used a lot of facial fillers in particular, let's say, as an anti-aging thing where there are wrinkles and for their face, they inject these kinds of gel-like material. Some of them, not all of them, a rare number of them have had some swelling at the sites on their face after receiving the Moderna vaccine. It is short-lasting, it goes away quickly, it's not a big deal, but it has been reported in the literature. So those are the only 2 differences that I can tell you. Outside of that, efficacy and, of course, safety seem to be very, very similar.
Thank you, Doctor. Next question is from Mrs. Rahimpour.
Thank you very much, Laupa, everybody, and thank you for the program. My question is, what about the mutation? When mutation happens, is the vaccine still effective? And the second question is, what is the percentage of false positive or false negative? Thank you.
Yes, so mutations are a concern, and the reason we keep hearing more and more about them is because there's more and more people on the face of the earth who have the virus, and so the virus has more opportunities to replicate, and during the replication, if it's a little bit sloppy in making its own edits of its own genetic material, it can have mutations. Many of those mutations will render the virus non-viable and non-virulent, and you will never hear about them. Of course, you only hear about the ones that tend to change things. And so currently, the two variants that have made a lot of news, one of them came from southeastern United Kingdom, and the other one came from South Africa. The United Kingdom one, so far, both Pfizer and Moderna have said that there is good efficacy with their current vaccines against it, that they are not thinking that this will be a game changer or will be a problem. The South African virus is interesting. Moderna had a study that they took 44 blood samples from recovered patients who had had the South African variant, and they saw that only 21 of them ended up binding with antibodies with this virus, meaning that just about half of them can escape immunity. So when they were calculating efficacy numbers, they thought that there's still some mild efficacy of the current vaccines, but it will be less than 50%, which is the mark that the FDA wants. So Moderna is already proactive about this. The beauty of mRNA vaccines is that you can edit the mRNAs the way that you have them and insert a new gene into them that is the recipe for this altered spike protein and include it in the future vaccines, although you still will have to go through a regulatory phase 3 trial again and present your data. So that's a 2-month event. But Moderna feels that there may be a need in the future for a third booster shot to try to cover for the South African variant if it were to become a very predominant form of the virus globally. But they're being very proactive about it. The UK variant, there's far less concern about it. Both variants are known to be more contagious, more likely to float in droplets in the air and be transmissible. So we have to be very careful. And there's now a recommendation that if you're in a poorly ventilated indoor space, double masking, because the number of layers in front of your face actually is helpful. It creates an obstacle course for the virus to get in, would actually be recommended. You talked about false negatives and false positives. This falls under the sensitivity and specificity of testing, of screening for anything that we have. Unfortunately, there's a very significant difference depending on whose test you're using, and also depending on what stage of disease. So, Mitra Khanum, if you were to mute yourself, please. What I would say that is that, for example, if you were exposed to somebody with COVID-19 yesterday and they call you today and say, look, you saw me yesterday, I'm COVID positive today, and you panicked and went and got tested, nearly 100% of the time your test will be negative. This does not mean that you're not going on to develop COVID-19 later. What it is that you happen to get tested at a stage where the virus had not replicated in your body long enough to produce copies enough to be picked up by the test. So early in the disease or early in the exposure, a lot of false negatives will happen. Once you start developing symptoms, with most PCR tests, both sensitivity and specificity are above 90%. With antigen tests, a lot of them fall in the 80s and the 70s. And so currently, because the virus is running so rampant in our communities, there is a recommendation that if you're having symptoms, you should assume that it's COVID-19 and start isolating yourself and start limiting any interaction with other people, even though your tests are negative.
Thank you, Doctor. The next question is from Kathleen Webster. Kathleen, go ahead.
Dr. Missaghi, I wanted to ask you, because I'm a little bit distrusting, and I wanted to know if you could explain why the protocol for introducing the vaccination was changed from all of the military being forced to take it to it's just those that choose to. That protocol changed. In the past, everybody had to take polio, everybody had to take smallpox vaccination, and the military were first and the Native American population second, and now they were not given first and second place. I didn't understand that.
I'm not familiar with this particular matter to really comment on it, but generally speaking, part of the distrust comes with coercion. And we now live in a different time, perhaps, that coercion by government is looked upon very unfavorably. So they want to tell people that we want you to voluntarily do it, especially if governments are forcing people to receive something. You know, private agencies have a lot more latitude in such matters. Like, for example, if I don't receive the flu shot every year, I can lose privileges at the hospitals where I practice. So I am essentially coerced to receive it, even if for whatever odd reason I didn't want to receive them. Or I would have to come up with some excuses like conscientious objection or whatever have you, or religious exemption. But because those are private entities and it's not government, they get to make it a condition of employment. Or you could have airliners who are private entities make it a condition of travel that either bring a negative test or bring evidence of vaccination so that they know that other passengers are safe. Once governments start in the business of coercion, I think it becomes a very messy situation from a human rights perspective. And so governments have been very careful not doing that. I think in some other cultures, perhaps in the East, maybe in China, there may be mass vaccinations with some coercions. I just don't think such a thing in North America would fly, or in Western societies with the current culture would fly.
Thank you.
Thank you, Doctor. Next question is from Blair's family.
Are you saying Blair Nichols? Okay. My question, first, thank you very much for being here and making all of this available to us. It sounds extremely reasonable to me. My question is, last Friday, I went to my chiropractor and their little machine that checks your temperature did not work. She still let people come in, and there were almost 12 people in that room, unventilated. And so then I said, are you going to have it working by Monday? I don't know. What does it matter? She said. I said, well, it helps me to feel safer in this situation. She said, well, if you feel good anyway, there's no problem. It doesn't make any difference if you have a temperature or not. I said, oh. And in my mind, I wasn't coming back. So is that a safe situation if the temperature doesn't work?
Yeah, so temperature doesn't catch everyone who is spreading the virus, but certainly if you have somebody with a temperature, you should avoid having them in enclosed spaces with everybody else. And so it's a very imperfect solution, but because it's a very cheap solution, and thermometers, especially ones that don't even have to touch the skin, that are, you know, thermal detection devices, can very quickly do this. I think it's very reasonable for any indoor space to check everybody's temperatures. As a matter of fact, there's now technology that automatically scans people and tells them to proceed if they have, they're trying to enter an enclosed space. The difference between enclosed spaces and open spaces is 18-fold I'm not talking about 18%, 18 times the difference of viral transmission. So if you had 5 people who were sitting on a patio around the table and enjoying dinner, as opposed to the same 5 people being indoors in a relatively small room, the chances of viral transmission that one of them, let's say, is an asymptomatic carrier is 18 times higher for the same time and duration of exposure. And so it's absolutely important to take every precaution, such as masking and distancing, but I think that temperature measuring is a very cheap and available technology, and I think that it behooves your chiropractor to invest that very small amount of money and ensure the protection of the patients.
You're mute.
Next question is from David and Kathy.
Professor Missaghi, from a world economic view and from a perspective of a world view in general, when we look at things that have been reported the other day, was one aspect that Africa is looking at not having any vaccines for until, you know, beginning or even middle of next year. And from the fact that we have a world economy that's, you know, affects everybody, you know, how do we as a I don't know what the word would be, but a world citizen, how would we affect getting those family members in Africa to get those vaccines? Is it possible to share the recipe? I mean, are there copyright laws or trade, you know, how does that work?
There is that's a wonderful question, and I wish that there was an easy answer to that question. I appreciate that you're thinking of this matter as a global problem, because it truly is. And even if you were to look past the unethical nature of countries that are less developed, having less access to this technology and this vaccination, you would still, only from the perspective of public health and epidemiology, would be correct to think that if you allow an entire continent or a very significant part of that continent not have vaccination, it would become a breeding ground for very significant mutations that will make their way back to your backyard and will create problems. One of the reasons why we think vaccination is the fastest and most ethical way to get to the end of this pandemic is precisely that if you were to allow the virus to run rampant in communities and just through natural immunity obtained from the virus itself, end the pandemic, you will have a very prolonged pandemic, and you will have a diverse number of mutations, some of which may be far more deadly, that will continue to pop up. And this cycle may continue so far that people who have even recovered may start losing their immunity, because the immunity obtained from the vaccine is actually so far proven to be more long-lasting than the immunity that's received from the disease itself, especially in people who've had it mildly, that their antibodies, in a portion of them, are already completely undetectable after 3 months. And so if you want to have that cycle going, it will continue to come back and bite yourself in the rear end by not attending to the less developed nations. So I think the economic forces of it, outside of even the humanity, will dictate that these that nonprofit organizations will have to partner with these governments and distribute vaccines far and wide. We already have had significant vaccine campaigns and significant medication campaigns against HIV, against malaria in the continent. And so I think with that sort of infrastructure that's already in existence, perhaps building on that and bringing the AstraZeneca vaccine to Africa would be in the works. The reason I say AstraZeneca, even though its efficacy is reported currently to be about 70%, it's a completely different technology, is that logistically, it doesn't need to be kept in these sub-zero temperatures, far easier to transport and far easier to mass produce and far cheaper. And so I think, and I'd said it before in one of my podcasts, that the AstraZeneca vaccine is likely the vaccine that will save the world, even though its numbers and efficacy are a little bit less than the mRNA vaccines in North America.
Thank you.
Thank you, Doctor. Next question is from Roy Rahimpur.
Laupa again, thank you. My question is, what percentage of people can have the virus but not having the symptoms? Is that possible to transmute the disease before having any symptoms of it?
Right. We don't know that just because the people who don't have symptoms tend to
You're muted.
we do not know what percentage of people can have the virus without having symptoms, just because people without symptoms typically do not seek care. But what we do know, the latest from the CDC, states that about 60% of new infections are occurring through asymptomatic carriers of this virus. And so they are among us. And so one of the things that I recently posted about was the first symptom of COVID is denial. You will have something very minor, like you're a little bit fatigued and you just blame it on having not slept quite well yesterday. You have one bout of diarrhea and you think, well, what I ate was probably just not quite right and it's going to go away tomorrow. You have the sniffles and you think my allergies are acting up, because they always do around this time of the year. And so you will always find some reason to deny the fact that something may be happening to your body. Given that we're living in the pandemic times, I think that you should take every symptom of your body very seriously and getting tested frequently when you have such symptoms, because what appears very mild in your body could be very deadly to your loved ones. And you just cannot say that, well, I have a mild version of it, you might as well get it from me and just get it over with. That does not work. There's no scientific basis to that.
Thank you, Doctor. And another question is from Mr. Chatirai.
Thank you. This was one of the best, most in-depth presentations I've seen so far. So thank you for doing it. Question I had for you is this concept of messenger RNA. I understand most of the vaccines, AstraZeneca excluded, are based on this idea of messenger RNA, where it tells the body what protein to produce. And in the case, in the specific protein they're trying to produce is something that looks like the spike capsule surrounding the virus, not the innards, but just the surrounding spike. It just strikes me as a computer scientist, where I'm trained about security ideas, like this is a massive vulnerability in the body. It seems like the body blindly produces whatever messenger RNA is presented to it. Doesn't this mean that there could one day emerge some kind of virus that uses messenger RNA in a negative way to produce bad proteins, if you will? Or how does the body differentiate between good or bad, or does it just blindly follow any messenger RNA that's presented to it? And how does it get into the cell in the first place?
So the injection, these are intramuscular injections. Thank you very much. And I think you are in Pennsylvania, because am I right?
No, we're in Canada.
Oh,
I used to be.
She used to be.
Oh, perfect. I recognized your lovely wife. So that's wonderful. Okay. I recognize that you're probably Canadian from your abode, but I wasn't so sure. So that's lovely. Thank you for the question. Yes, so messenger RNAs are just recipes for proteins. Proteins per se are not really dangerous. And when the body sees a protein that it does not recognize as self, it typically develops a reaction to it. And that's what we want with this vaccine. Could there be some sort of a protein that could be bad for you? The only protein, because protein is different than genetic editing and all of that. Genetic editing requires enzymes. It's a far bigger production than this particular thing that we're talking about. But to just give the body the recipe to make a particular protein, the only place that I see a vulnerability is with prions. This is a little bit beyond the scope of this conversation, but prions are the causative agent of mad cow disease or kuru, which comes from Papua New Guinea. That they used to be called very slow viruses because they thought these are viruses, but they truly were proteins that somehow, when they were entered the body through ingestion of the brains of an affected animal, or an affected human, in the case of Papua New Guinea and Kuru, they would then find similar proteins and start changing the conformations of those proteins in the body. And over a 10 to 20 year period, these patients would develop severe neurologic manifestations because it would be very neuroinvasive. So I guess potentially you could have a really bad acting protein that you teach your body how to make, and then it could wreak havoc in the body itself. Nonetheless, short of something like that that could be done maliciously, I suppose, any other protein is really just a protein, and proteins have different conformations and different shapes. Some of them are immunogenic and some of them are not, and they're just simply digested. And this particular spike protein that we make, believe it or not, doesn't even last in our system that long. Our system will digest it. By digest, I mean we'll break it down. It's not through the digestive tract here. We'll break it down and it will actually destroy what it has created by itself. How does it get into the cells? When you inject into the body of the muscles, some of it will end up in the cytoplasm of the cell because of just the way it's formulated in this lipid emulsion at the cold temperature that it is. And very small amounts of it need to get into the cells. And so the process of what we call translation means you go from mRNA to protein, happens at the level of the cytoplasm, where you read the instructions, the recipe, and you produce the protein. And then that is where the immunogenicity also happens, because this protein is produced and then the protein ends up looking as non-self. The body doesn't recognize it, so it activates the immune system to come look at it, and basically in the process of trying to destroy it, it ends up making antibodies to it.
Thank you.
My pleasure. All right, well, I think that we've had a very long night, and I apologize if I'm long-winded. Brevity is not one of my strong suits, but if there's another question, we'll take it, and then perhaps we will adjourn the meeting. Do we have any other thoughts? I can't see all of you on one screen.
I sincerely appreciate Dr. Missaghi for his time. I really appreciate it. And we are so proud to have him in our community.
You are very kind to me. I am also very indebted to you with all of the wonderful efforts that you do in cultural activities and bringing people of various walks of life together, from hosting of Spirituality Cafe to hosting the Hamzaban Foundation events. Your team, Dr. Hajbande, has been very instrumental in bringing so many people together and promoting understanding and knowledge. So thank you for all that you do. And also my gratitude is due to the Baha'is of Surprise, Arizona, who put together this wonderful gathering. I am sorry if I was long-winded, and I look forward to more discussions. I think that time, as far as time, we can say that more of the pandemic is over and less of it is left. But in terms of the human toll, we can say that the worst of the pandemic is probably ahead of us, given the current numbers and the difficulties with the waves that we have. So
I received a comment from one of the audience that this was an excellent presentation, and he recommended that next time we should advertise for it more extensively than we did this time. That person is right. We should have done more advertisement. Thank you very much.
And also, there is another hand up.
Okay, David.
I was just going to ask if it's possible we can find out how to receive this video that's being recorded to share it with others.
Actually, I just want to mention that since this was one of the projects of the Spirituality Cafe, there is a very strong possibility that we can make it available through YouTube and podcasts. So as soon as the committee decided how it could be distributed, the friend will be informed. And that shouldn't be that long, maybe within the next couple of weeks.
Okay, so perhaps anybody who wants to receive it, post your email in the comments section, and I'll have Mr. Mozaffari record those emails to ensure that once this is published in any form, those emails can receive the link to the presentation if they wish to receive it. Friend, I am going to excuse myself. Thank you very much for all of your attention, and I appreciate your patience with me. Thank you so much.
Thank you, Dr. Missaghi. Thank you very much.